Dear students, now I am going to talk about another method of introducing cloned genes to host cells, transduction. So, you know, gene delivery or the process of introducing foreign genetic material such as DNA or RNA into host cell. So, gene delivery must reach the genome of the host cell to induce gene expression. So, whatever the foreign gene we have introduced into the host cell incorporate it and it has to express it. So, that is the purpose of introducing cloned genes to the host cell. So, gene transfer to the cell brought by several methods. You have studied already physical methods such as particle bombardment or gene gun method and electroporation, chemical methods such as liposome mediated gene transfer. Today I am going to talk about the biological method of gene transfer, that is transduction. So, transduction is the natural process by which a virus transfers genetic material from one bacterium to another. Here you will see how exactly a bacteriophage, or this is bacterium. So, this is a bacteriophage. It has infected this bacterium and they are introducing their genetic material into the host cell, that is the bacterium. So, viruses called bacteriophages infect bacterial cells and use them as hosts to make more viral particles or viruses. Scientists have taken advantage of this transduction process to introduce a gene of interest into host cells using viruses as vectors. So, this in the genetic engineering, this process is exploited to introduce the large gene of eukaryotic cell to the host cell. So, here the viruses, they have engineered to introduce the large genes and this virus made to infect the host cell. So, here the virus is not causing any disease or discomfort to the host cells, only it is used as a gene carrier. So, this method involves the introduction of genes to host cell genome using viruses as carriers. So, transduction is a biological method of introducing foreign DNA or RNA into bacterial or eukaryotic cells using viral vector. So, you will see in this cartoon how exactly this is adenoviral vector. So, it has emptied its DNA, or else to its own DNA we can introduce our gene of interest and made to infect the host cell and it get into the host cell nucleus and bring the stable transfection or else transduction. Viruses are used as vector to transfer gene of interest to host cell. Replication deficient, genetically engineered. So, these are replication deficient. So, they cannot multiply, these viral particles cannot multiply in the host cell, only they are used as a carrier of our gene of interest, or else they are introducing our gene of interest into the host cell. They cannot multiply inside this. So, replication deficient, genetically engineered recombinant viral vector carrying gene of interest made to infect cells. So, this for example, this may be carrying our gene of interest, insulin producing gene. So, they are introducing this gene to the host cells. So, these viral particles, they cannot multiply here. So, only they will be our gene of interest will be delivered to this host cell. So, viruses are used in gene transfer due to following features. So, the main feature is efficiency of the viruses to deliver the nucleic acid into the cell. So, that is how the viral particles are efficient to introduce the genetic material into the host cell. Then another, high level of replication or gene expression. So, whatever the DNA introduced or genetic material, gene of interest introduced into the host cell, they have a high replication efficiency. So, that's why this method is very popular. So, bacteriophages bacteriophages, the viruses infecting the bacteria, adenoviruses, retroviruses, the coronavirus, retroviruses, RNA is the genetic material, herpes simplex as the virus, herpes viruses, bovine papillomaviruses. These are all the popular viruses. They are used as gene carriers, or else they are used to transfer gene of interest to the host cells. These recombinant adenoviruses are used to transduce many mammalian cell types, so such as hepatocytes, or else liver cells, neurons, then muscle cells. All these cells are their gene transfer can be efficiently done to all these cells using the recombinant adenoviruses. High titers, that is the high amount of adenoviral vectors, vectoral solution, have high transducing efficiency. So, whereas retroviral vectors bring stable integration of gene of interest. So, these are all why several viral vectors are developed. So, adenoviral vectors they can be introduce our gene of interest to different types of cell types, whereas retroviral RNA viruses they can bring stable transfection or stable integration of the gene of interest. Transgene or gene of interest may be incorporated into the viral vectors either by addition of the whole genome or by replacing one or more viral genes, which can be achieved by ligation. The process of ligation brought by DNA ligase enzyme. So, it is introduced into the whole genome or else replacing some viral genome elements, and so this is different method of transduction. Gene of interest can be introduced into the viral vectors. So, the foreign gene is packaged into the viral particles to enter the host cell. So, these are the viral vector, it is packed with the gene of interest, and this is going to infect this is the host cell. The entry of viral particle containing gene of interest into the cell and then to the nuclear, first it get into the cell cytoplasm, then it is get into the nucleus and incorporate the gene of interest into the host cell genome. So, the attachment of the viruses mediated binding of the receptors, it may bind to the receptors and getting inside through endocytosis. Virus reach the host cell nucleus and inject the DNA of interest into the host cell nucleus. Here in this is a retroviral. Here you can see it is sending its RNA. The RNA is converted into DNA. This DNA get into the host cell genome and it get incorporated into the host cell genome and get transcribed and get expressed. So, this is a therapeutic protein get expressed. This is how transduction is used to introduce gene of interest to the host cell. The vector genome remain as an episome. It will remain, it may not be incorporated into the host cell genome. ಅದು ಹೋಸ್ಟ್ ಸೆಲ್ ಜೀನೋಮ್ ಜೊತೆಗೆ ಇನ್ಕಾರ್ಪೊರೇಟ್ ಆಗದೇನೆ ಸಪರೇಟ್ ಆಗಿ ದಿಸ್ ರಿಮೇನ್ ಎಸ್ ಎ ಎಪಿಸೋಮ್, ಹೌ ಎಕ್ಸಾಕ್ಟ್ಲಿ ಪ್ಲಾಸ್ಮಿಡ್ ಇನ್ ಎ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್. ಸೊ ಆರ್ ಎಲ್ಸ್ ಗೆಟ್ ಇನ್ ಟು it may be incorporated into the host genome or host DNA, okay, and start expressing the gene of interest whatever incorporated. After entering into the nucleus, the viral DNA remains epichromosomal. So, it may remain as a separate epichromosomal gene or else it can integrated into the host chromosome and therefore does not activate or inactivate host genes. So, the host genes are not get affected by this transduction method. It is only we can deliver our gene of interest into the host cell without affecting the host cell genome. Transduction method of introducing gene may result in transient expression of the host cells, or else they may be expressed, prolonged expression can be achieved in the case of the cells, non-dividing cells like neurons. If we incorporate, we can make this gene expression for long duration. If it is sometimes if it is actively dividing cell, the gene expression will be short living. After some generation, the cells may lose our gene of interest, whatever introduced through this transduction method. So, what are the applications? So, the engineered retroviruses are used as a vector in gene therapy or to transfer gene to cultured mammalian cells. They can accommodate 90 kilo base pair size desired gene we can load into such viral vector. These are used in gene therapy of the SCID. SCID means it is a type of immunodeficiency. It is this combined immunodeficiency syndrome. So, where you can see the person not having any immunity to any of the infection. So, this is what you can see then or else hyperlipidemia. So, all these genetic disease gene therapy can be done efficiently with the help of retroviral vector. Adenoviruses are also used as a vector in gene therapy in liver cells, lung, muscle, heart, central nervous system. These viral vectors are taken up efficiently by the cells in vivo. So, it is in vivo we can do the gene therapy with the adenoviral vectors. The adenovirus derived vaccines have been used in humans without any side effects. So, the adenoviral vector have high cloning capacity. They can take up 37 kilo base pairs and high transduction efficiency and long term transgene expression and lack immunoresponse or cytotoxicity to the host cells. They are not affecting the host cells. So, host is not getting any immune response due to this adenoviral infection. So, the lentiviral vectors used in gene therapy of hematopoietic cells such as during blood cancer and all, then muscles, neurons, hepatocytes, all these gene therapy can be used where you can see lentiviral, engineered lentiviral vectors. Through transduction, transgenic embryos can be produced, transgenic animals are produced, undifferentiated embryonic stem cells are inserted with gene of interest, foreign DNA through viral vector through transduction, then transferred to blastula. So, the blastula carrying recombinant cells is then implanted to the uterus of the foster mother to get transgenic animals. So, this is a popular method of producing transgenic animals through using transduction technique. This technique is very useful in the study of genetic control of developmental processes. So, in the research also this method is used. So, this is here in the animation how you will find a bacteriophage introducing our gene of interest into the host cell. Thank you.